Hello, everyone. I'm Rick Lifton, president of the Rockefeller University, and I'm delighted to welcome you to another of our sessions, Virtual Discussions with Genuine Experts. This is our first session in 2021. Today's speaker is the remarkable virologist Paul Bieniasz, whose research has transformed our understanding of viruses and their interactions with human hosts. Over the last 11 months, our nation and the world have experienced the tragedy and continuing challenge of a global pandemic of staggering proportions. More than 100 million people have been infected with the SARS-CoV-2 virus, the cause of COVID-19, and more than 2 million people have died from complications of this infection. The US has been particularly hard hit, with more than 27 million cases and over 460,000 deaths. The global response to the pandemic has had many dimensions: public health efforts, clinical efforts to figure out what works to keep people alive, along with politicization of the prevention effort. But without doubt, the world has benefited mightily from the foundation of basic science that has fueled the rapid identification and characterization of the virus that causes this disease, the development of tests for the virus, the identification of the virus's Achilles heel, affording efforts to develop new therapeutics, and the unprecedented development of highly effective vaccines in less than a year, a truly remarkable accomplishment. These vaccines can prevent up to 95% of symptomatic disease and nearly all severe disease, including cases in individuals at high risk of severe disease and death, which increases dramatically with age beyond age 50. These highly effective vaccines are now being distributed and provide a crystalline example of the enormous impact of fundamental basic science on human health in response to an extraordinary public health emergency. And yet, just as these vaccinations are scaling up, basic science enabling the rapid sequencing of the viral genome to monitor them for emergence of variant viruses has identified a growing list of variant viruses that threaten the efficacy of vaccines and therapeutic antibodies. Rockefeller scientists have played critical roles in the global response. Today's speaker, Paul Bieniasz, has been one of the leaders in this effort. Along with many other Rockefeller investigators, he was able to hit the ground running early last year thanks to years of foundational basic science conducted in our laboratories. In fact, Paul, his wife, Theodora Hatzionu, Charlie Rice, Marina Kasky, and Michelle Nussenzweig have led research that has identified key targets on the viral spike protein for antibodies that prevent viral infection, and have identified mutations that have predicted how the virus will evolve to escape these antibodies. And in recent months, as we've begun to see the emergence of variant viruses that have acquired mutations that may reduce the efficacy of these vaccines, Paul and his colleagues have tested how well these vaccines are likely to fare when they encounter these variant viruses. The current uncertainties demonstrate that we will likely need a range of therapeutic and preventive agents to finally bring this pandemic under control. We are grateful to all of our benefactors, past, present, and future, whose support has been critical to advancing the research that will get us out of this pandemic. Your support is vital to our efforts to battle COVID-19 at an accelerated pace. Before turning the podium over to Paul, I will give you an update on the current state of the COVID-19 pandemic. This is the current state of how many cases there are every day. And as you can see, starting back last February until now, cases have had quite a variable rise, a few peaks occurring along the way, and then a dramatic acceleration in the fall with a bump after Thanksgiving, another bump after Christmas, hitting a peak of more than 800,000 cases per day worldwide. And now we're in the midst of a brisk reduction in cases over the last month. To date, there have been more than 106 million cases worldwide and more than 2.3 million deaths. In the United States, Similarly, there has been a lot of activity in a lot of cases, again, in 3 distinct waves. Compared to our last webinar in December, where we were concerned that we were headed toward 200,000 cases per day, we accelerated right through that by Christmas, hitting over 300,000 on a single day with a peak of about a 7-day average of about 250,000 cases per day. But over the last month, there has been a steady decline in the total number of cases, with a daily average now of about 125,000 cases per day. Nonetheless, the number of deaths per day has continued to rise. And while in December we were lamenting having a peak of nearly 2,500 cases, approaching 3,000 cases per day, we actually hit over 4,000 cases per day by the time we hit the beginning of the new year. And we're still continuing with a very high death rate across the country. Globally, the pattern is complicated. It's quite variable and heterogeneous across the globe. The United States continues to be among the most active areas with the prevalence of virus worldwide, although it's been improving as we discussed. Still parts of Europe with very high levels. Israel, despite a vigorous vaccine campaign, still has very high levels, but they're beginning to come down. And it continues to show this pattern that we've commented on in the past, where Southeast Asia and very large parts of Africa appear to be relatively spared of cases and deaths from the virus. This is what the distribution looked like across the country last December. It rose to a much worse pattern than this by the middle of January, but has now been coming down substantially to a pattern that looks much improved lately. And this obviously has been welcome news. This is what the pattern looks like in New York City, where you can see that we hit extraordinary highs in the beginning of January and have been slowly starting to come down. This apparent drop is attributable largely to the snowstorm last week that dropped the number of cases that were, the number of tests that were being done on a daily basis, and we're still clocking along at over 4,000 cases per day in recent days. We still have a significant number of new hospitalizations that's just barely beginning to tail off, and the number of deaths per day is still relatively steady at about 75 deaths per day in the city. As we've discussed previously, the vaccines target the entry step of the virus getting into the cell. This occurs through the viral spike protein, which interacts with the ACE2 receptor on the surface of some respiratory epithelial cells that allows it to gain entrance into the cell and go through its life cycle and then gets budded and released into the circulation to infect other cells. So this has been the target for the vaccine, all of the vaccines that are in clinical trials or beyond. A wide variety of approaches to the vaccine have been taken. These have included inactivated or attenuated viruses that have been pursued by several Chinese companies, peptide vaccines, and Novavax released early results from its phase 3 clinical trial. Engineered viruses, predominantly adenoviruses, and we now have reports from Johnson & Johnson, the Russian company Gamaleya, and Oxford AstraZeneca. And we now have 2 approved RNA vaccines, which had never been done before. And these are the RNA vaccines from Pfizer and BioNTech, which we talked about last time, and Moderna's RNA virus, which is very similar, which reported out and has been approved by the FDA for emergency use. This is the data on the Moderna RNA vaccine, which is newly approved since our last meeting, and it looks very similar to the Pfizer vaccine. One of the main differences is they spaced their doses of vaccine by 28 days rather than 21. So after the first dose, you can see that starting about day 14 or 15, after the first dose, before the second dose, the curves start to part, with the number of cases in placebo rising, but the number of cases in the vaccinated group staying relatively flat. And by the end of the trial, after the second dose, they ultimately had 185 cases in placebo and only 11 cases in people who had been vaccinated, 30 severe cases in placebo and none in the vaccinated group. So the efficacy for prevention of symptomatic COVID-19 was 94%. Extremely effective, very similar to the performance of the Pfizer vaccine. More recently, the Gamaleya adenoviral vaccine reported out results of its phase 3 clinical trial, and this has now been published last week in The Lancet. 22,000 people, a two-dose vaccine. They had, I think, the best approach at an adenoviral vaccine. The concern with adenoviral vaccines are you give an adenovirus once, the immune system is stimulated, learns to recognize not just the viral proteins from SARS-CoV-2 that you want it to, but it also recognizes the adenoviral proteins and makes it harder to come in with a second dose and not have the immune system react and diminish the efficacy because it gets cleared before it can infect any cells and give you any added boost. So Gamaleya had the good idea of using two different adenoviruses, adeno 26, which is people are rarely exposed to this virus in the human population. Adenovirus 5, in contrast, many people have been exposed to the virus. About 40% of Americans, some parts of the world, up to 70 or 80% of people have circulating antibodies. But the thought was that if you come in with this virus second, if you haven't seen the virus for a long time, the immunity may be waning and you can get a good boost from that. I'll just mention that the other adenoviral approaches have used the same adenovirus for both dose 1 and dose 2, which is an interesting strategy that I don't think was likely an optimal choice. And people have been rethinking whether you want to be doing two doses with the same adenovirus. And this may have in part motivated Johnson & Johnson's goal of seeing what they can do with a single dose of vaccine. In any case, the Gamaleya phase 3 clinical trial was all done in Moscow. Similar outcomes of scoring symptomatic infections, and this shows the departure of the curves of placebo and vaccinated individuals. And this looks like a very successful vaccine as well, with about 92% efficacy. Consistent across all the ages studied, although this was a somewhat younger population than in the Moderna vaccine trial, where about a third of the population was over age 60. But they also had no moderate or severe disease among the vaccine group. This is a very good result. So we now have two vaccines that are registered in the United States, the Pfizer and the Moderna vaccine. We believe that per the administration's recent efforts, they now have secured commitment to 300 million doses of both the Pfizer and Moderna vaccines by the end of summer. I haven't seen an official statement that that's been solidified, but since the end of January, that was what the expectation was. And of course, when you do the math for a two-dose vaccine, together you could vaccinate 300 million Americans, which obviously would cover virtually the entire adult population. There have not been clinical trials to date on people under age 16, and most trials have started at age 18. So there's very little experience in the pediatric population to date, but now companies are beginning to pursue trials in that population as well. Johnson & Johnson has filed for registration. I'll come back to their phase 3 trial in a moment, and Novavax has reported out in the interim as well. So this shows how the vaccinations have been rolling out in the United States. This is a slide from Bloomberg. And obviously we had a kind of a rocky rollout with very uneven distribution and delivery of vaccines. This has been smoothing out over the last several weeks, and recently hit a high of 2 million doses delivered in a single day. And if you do the math, you're going to need to pace at least that much and likely a bit more to get to complete delivery of 2 doses to 300 million people by the end of the summer. But that's a laudable and I believe worthy goal to be shooting for at this point. Obviously, the manufacture and distribution and delivery remain logistical challenges at every point along the pipeline. But these vaccines are starting to roll out worldwide, and Israel has been the first to really get going at a high level. They now have 62 doses per 100 people in the population, and have 23% of their population that have been fully vaccinated. Almost all of these are people over the age of 60. And this is, in fact, a high fraction of that population. And as a consequence, investigators have been able to look at the infection rate in that population before and after vaccination, and also comparing infection rates in that population compared to the change in infection rates of younger people, and have shown a clear difference demonstrating clinical benefit in the general elderly population of getting vaccinated. And this is obviously extremely good news. The United States has delivered more vaccines than anybody else and is doing quite well at the international level in terms of getting people vaccinated as a fraction of the population. So the fly in the ointment has been, since we last met in December, several variant viruses have started to emerge. This is not altogether a surprise, but it obviously is worth noting and is of concern. Why should we be concerned about variant viruses? Viruses, as they replicate, acquire mutations all the time, and most of these are going to be random occurrences that are going to have no functional consequences. But if any of these mutations confer an advantage to the virus, either in its ability to survive, to get out of the cell and propagate, or to increase its ability to infect cells, those changes will be selected for, and if they have these advantages, they could take over the population of viruses that are circulating in a population relatively rapidly, and could make it tougher to fight off the virus with your own immune system, but also in particular could be more resistant to the existing vaccines, or for people who have previously been infected to be able to reinfect people, because the virus may now be immune to the immune system response that a person made when they were originally infected. So these are obviously matters of great concern to be followed. As you may know, some viruses are very successful at evolving in this way, such as the influenza virus, and that explains why we need to get an influenza booster every year, because the virus has changed substantially from year to year. But other viruses do not have that facile ability to evolve rapidly. The measles virus, for example, we give that once, and we typically have lifelong immunity to the virus. So these mutant viruses are rare in occurrence, but if multiple changes that collectively may be beneficial to the virus can occur, particularly if the virus is allowed to replicate in a host for a prolonged period. So imagine, for example, somebody who has a compromised immune system who can make some antibodies to the virus, but can't make enough to clear the virus. That allows, in a single infection, the virus to continue to evolve in a patient and acquire many mutations, some of which may be beneficial to the virus, and some of which in combination may be particularly beneficial to the virus. This is how these viruses can go from having no useful mutations potentially to having several mutations that benefit the virus's ability to avoid the immune system and or to gain access to cells through increased infectivity. So there are three of these variant viruses that have been emerging over the last couple of months. One of these is the B.1.1.7 virus, which has gained a toehold and has increased in frequency dramatically in the United Kingdom. So this is the genome, the RNA genome of the coronavirus. And as you can see, the mutations are not distributed. So the mutations are shown as these spokes of the wheel. And as you can see, they are not distributed randomly across the viral genome. In fact, they are highly clustered, predominantly in the spike protein, which is the protein that gains access through binding to the receptor to get entry into cells, and a couple of other proteins that are derived from the viral RNA. So these mutations in the spike protein are particularly noteworthy and concerning. And Paul Bieniasz will talk about the impact of some of these individual variants in these viruses. So this is the virus that is now predominant in the United Kingdom. There's a second virus that is of concern, and that's a virus called B.1.351, and this is a virus that has virtually completely taken over infection in South Africa. And again, you note that there are very few mutations anywhere in the genome except in the spike protein, and these are clustered in the spike protein in this case. And it is noteworthy that there is one mutation that is shared between the mutation found in the UK strain of the virus and the South African strain of the virus. This is quite unlikely to occur by chance and suggests that this variant may be playing a role in giving this virus an advantage over other viruses circulating in the population. And then the third virus that has emerged to date is prevalent now in Brazil, and this is the P1 coronavirus. And again, you can see this clustering in the spike protein, again, not likely a chance event. And in particular, you will note that there are, in this case, three variants. This mutation that alters lysine at position 417 to threonine, a glutamate to a lysine mutation, and an asparagine to tyrosine mutation. These three variants are also seen in the variant that is circulating in South Africa, and yet there does not appear to be a common ancestor between these two viruses, suggesting that these have emerged independently on these two lineages and are extremely unlikely to have occurred by chance and are likely playing an important role in development of the disease in these individuals. So these emergent viruses are clearly of concern, and we're starting to gain some clinical experience, which augments the work that Paul and his team have done in evaluating the impact of these biochemically in the laboratory. But these three variants cluster. Now we're looking from the top down on the top surface of the spike protein. And the spike protein is a trimer. It has three subunits of the same protein. And you can see, for example, the E48K variant is present at 3 locations, as are the other 2 mutations in the protein. So these are right at the surface of the protein where the virus is binding to its cellular receptor to make contact. And there is strong suspicion that these are altering that interaction. And maybe masking the ability of antibodies to recognize these particular sites, particularly in people who have previously been exposed to the virus that does not have these mutations. So this has been looked at already in the trial released just in the last 2 weeks from Novavax. So this is the peptide vaccine that tested vaccine versus placebo. We haven't seen anything other than the press release from the company. They studied 15,000 subjects in the United States. Many of these were over age 65. It was a 2-dose regimen. At the first interim analysis, they showed a 90% reduction of symptomatic infection in the vaccine group, a fantastic result, particularly when you consider that half of their patients had the UK variant of the virus. And in fact, in the people who had the UK variant, they had about 86% efficacy for reduction of symptomatic infection, whereas the people who had the classic version, if you will, of the virus had about 96% protection from infection. So this is clearly very effective on these viruses. However, they also did a study in South Africa where they studied 4,400 subjects, and they found a reduced efficacy of their vaccine. It only provided a 60% reduction in symptomatic infection in their vaccinated group that was not infected at the same time with the HIV virus. And in this population, as expected, the vast majority of infections were with the South African variant virus that I've described to you. So this is a strong indication that this vaccine is highly efficacious, quite similar to the Moderna and Pfizer vaccines on the older versions of the virus, but on this South African virus, it had markedly reduced ability to prevent symptomatic infection. We don't know with certainty how well it worked to prevent severe disease or death because most of these subjects in South Africa were young and were not predisposed to developing severe disease. Johnson & Johnson similarly reported out results of their trial of vaccine versus placebo. As we discussed, this was an adenoviral vaccine where they used a single dose of the adenovirus 26 viral vaccine. Again, this vaccine carries the spike protein of the SARS-CoV-2 virus inserted into this viral capsid. Again, about a third of the patients were over age 60. And 28 days post-vaccine, they found protection against moderate to severe infection of 72% in the United States. But they also did a South African cohort, and they found 57% protection in South Africa, and they found 66% protection in another cohort in Latin America. So they had about 20,000 in the US, 18,000 in South America, and 6,500 in South Africa. They reported an overall 85% reduction in hospitalization. They did not fractionate that, so we don't know how that looked in South Africa. But again, likely a younger population in South Africa, but I have not seen that data reported. But once again, you see that the single dose of this adenoviral vaccine was quite effective, not as effective as the Moderna and Pfizer and other vaccines, but only 57% reduction in South Africa. And we don't really know with certainty what effect it had on reducing hospitalization and severe morbidity or death. So this is where we are with the vaccines. So the great news is there are a number of efficacious vaccines. The serious questions and concerns are that we have these variant viruses that are starting to spread around the world. The UK variant now is the predominant variant in the United Kingdom, less so elsewhere. There are now cases across most of the US. The South African virus is all over that country, has spread less through the rest of the world thus far. This appears to be a more worrisome virus, but we don't have enough data to know what impact it has on whether people will get infected. Will they get severe disease with this virus much more than they would have with prior iterations of the virus? What will happen with further dissemination of this virus? Will it overtake other countries as well, or will it not be able to get a toehold? And we don't know much about what the course of this will look like thus far, particularly when you have these different viruses competing with one another in the same population. There have only been a handful of cases reported in the United States thus far, but we haven't been sequencing viral genomes that much in the United States. So we really don't know how extensive the spread of that virus or the Brazilian variant of the virus has spread into the population thus far. So a lot of questions that we'll get to momentarily when we turn to Paul, but I do want to give a couple of updates of other research from Rockefeller Laboratories since our last connection. In particular, I've told you previously about monoclonal antibodies that have been developed by Michelle Nussenzweig and many colleagues shown here on this slide. This has progressed to first-in-human trials. They're a combination of two monoclonal antibodies derived from the genes that encode these antibodies that were taken from patients who had recovered from SARS-CoV-2 infection. The two that have been put into clinical trials are by far the most potent of these monoclonal antibodies. And one of the possibilities that is being looked at is whether these monoclonal antibodies might be efficacious when given as subcutaneous injections rather than intravenous infusion. The uptake of the existing monoclonal antibodies has been limited because of this need for intravenous infusion of the antibodies for either preventive or therapeutic purposes. And this has been a limitation. And we will see whether these new monoclonal antibodies have efficacy in a subcutaneous formulation. And also will be critical for these monoclonal antibody therapies to know how efficacious they are in confronting these variant viruses that are now circulating in the population. Other work that I'll mention briefly, it's been recognized that there's a need for other therapeutics for these viruses. Orally available drugs would be very advantageous. If you get infected, you give it a couple of pills and you start on treatment immediately. And there are efforts to pursue novel targets for this. Tarun Kapoor has worked on an essential enzyme required by the virus for its function. And this is an RNA methylase that caps the RNA and protects it from degradation. And Tom Tuschl has worked on, he's worked on the helicase, which unwinds the viral RNA after it's been replicated. And Tom Tuschl has worked on the methylase. And both have identified promising leads that have gone into medicinal chemistry at the Tri-Institutional Therapeutic Discovery Institute. That's a shared resource of Rockefeller, Sloan Kettering, and Weill Cornell. And Bob Darnell, I should mention, has developed a spectacular, very inexpensive, very accurate, rapid test for the virus that we've had in use here at Rockefeller for the last 7 months. And we currently are testing everybody on campus once a week to make sure that we do not have any viral spread occurring on our campus, which I'm happy to say we do not have. So it's been extremely useful to us. It has not been lost on Bob and others that this could be a very useful test in other settings as well. And in particular, as we've debated in New York City, how do we keep the schools open when we have children who aren't going to be vaccinated anytime soon? One possibility would be to have more aggressive testing programs. Of course, having a test that is very inexpensive and very easy to administer. Bob's test is a saliva-based test, doesn't require a healthcare worker to take a swab up your nasopharynx, and it's a very attractive option, and this is being explored now. I will now come back to Paul Bieniasz. So I've already told you a little bit about Paul and his work. He leads Rockefeller's Laboratory of Retrovirology, and we're proud that Paul is also one of the nation's Howard Hughes Medical Institute investigators. Paul has dedicated his career to reducing the human toll that's exacted by infectious diseases. Educated in England, Paul came to the United States in 1996 to do postdoctoral research at Duke University. And 3 years later, he was recruited to New York to join the faculty at Rockefeller along with the Aaron Diamond AIDS Research Center. And he was promoted to Rockefeller Professor in 2010. Paul has been a leader in HIV research, identifying mechanisms by which HIV overcomes cellular defenses, revealing potential targets for therapeutic intervention. He and his wife, Theodora, who I've mentioned already, expanded the possibilities of HIV research by creating altered forms of HIV that can be used in animal models to accurately mirror infection in humans. Paul's team has since developed increasingly sophisticated techniques to manipulate viral genomes. So when the pandemic hit, Paul and Theodora quickly pivoted to apply their expertise to the study of COVID-19 and have done crucial research that has made key contributions to both their own work and collaborative research with other investigators at Rockefeller and at the New York Blood Center and across the city. Paul's lab has made many contributions to our understanding of how the novel coronavirus infects human cells and how the human immune system responds to infection. This has made Paul a go-to expert for journalists reporting on the evolution of the virus and on the biology underlying vaccine-induced immunity. His research has been vital in identifying the steps we can take to prevent and treat new infections in the ongoing pandemic. So I'm now pleased to turn the podium over to Paul. At the end of his presentation, we will answer your questions. You're welcome to submit questions at any time using the Q&A function at the bottom of your screen. And now I'll turn it over to Paul.
It's my pleasure to join you this afternoon. Thank you very much for the introduction, Rick. And what I'd like to do is give you a sort of an update of what we've been studying in terms of antibodies and SARS-CoV-2 in the context of what we've just heard about, the vaccines that have been deployed and the new variants of the virus that we're beginning to see circulate in various countries around the world. So Rick already covered some of this, but let's just we're going to drill down a little bit in some details as we go through the biology of the spike protein and the antibodies that recognize it. So as you heard, the spike protein decorates the exterior of the virus particle. It's the key part of the virus that touches the surface of the cell and allows the virus to enter into the cell. And it's also a key target, the key target really, of the antibodies that provide protection either from reinfection or after vaccination. And so as Rick mentioned, it's a trimeric protein. So one of these three subunits is highlighted here in color. There are really two parts of it that are extremely important for the recognition by antibodies. The NTD, or N-terminal domain, is a target for some quite potent antibodies, but really the most important target for really the most potent neutralizing antibodies is this RBD, which is the receptor binding domain. This is the part of the protein that flips up, as you can see in this blue blob here, that interacts with the ACE2 receptor. And many of the antibodies that are neutralizing, that are most effective at interfering with virus replication, bind directly to the RBD and block its interaction with the ACE2 receptor. Okay, so before we get into the details, we need to have a little bit of background on B cells and antibodies, how they are made, and a little bit of terminology. So circulating in our blood and lymphoid tissue are B cells. There are tens, perhaps hundreds of millions of different B cells, and they're different because they have different, what we call B cell receptors on their surface. Now, when we're infected by a virus, for example, SARS-CoV-2, a small subset of those B cells, perhaps tens or hundreds of them, will have the ability to recognize the proteins on the outside of the virus particle. In this case, the spike protein is what we're particularly interested in. Now, that recognition event triggers something in the B cells. They grow and divide. They grow and divide, and after a while, they stop growing and dividing and start to make antibodies. Now, during this process of growth and division, the B cells, they are essentially selected for the ones that are best able to interact with the viral proteins. And as part of that selection process, the antibody genes themselves become mutated. And they are selected for higher, tighter binding to the viral protein. So the antibodies that are eventually made are a little different to the B cell receptor genes that are initially displayed on the B cell surface. They have been mutated, matured, as we call it, into forms that bind tighter to the viral protein. And this is a really quite important feature of antibodies. Now, once these antibodies that are, as I say, directly derived from these B cell receptors float around in the circulation, if there are viral particles around, they can, just as they did on the surface of the B cell, interact with the viral particle, and a subset of them, what we call neutralizing antibodies, will then prevent that virus particle from being able to infect cells. Antibodies work in a second way. If the cells manage to get past this first line of defense and the cells do in fact become infected, the antibodies can recognize the very same protein, the viral spike protein, on the surface of those infected cells. And a cell that's coated with antibodies like this serves as a flag to other cells of the immune system to come and kill and eat that cell. So antibodies really work in two major ways. One is by neutralizing virus particles. The other is by targeting virus-infected cells for death. And both mechanisms are very important. The neutralizing activity of the antibodies is perhaps especially important, and it's really especially important to us because we can very easily measure it in the laboratory. Okay, and we can show that ability to neutralize is really a very important component of how antibodies work. Okay, so over the last almost a year now, we've been working very closely with a team at Rockefeller, in particular Charlie Rice and Michelle Nussensweg, to really understand the nature of that antibody response that I've just told you about in the context of SARS-CoV-2. And this rather intriguing diagram that you can see on your screen now is sort of a representation of a large part of that work. So each one of these slices of this circle, or pie as we call it, represents one individual. And those individuals labeled in green those are people who have had SARS-CoV-2 infection. Everyone else represented here, these individuals around here, they are people who have been vaccinated either with the Pfizer or the Moderna mRNA vaccines. And these slices represent antibodies, okay? Within each slice is represented several tens up to 100 or 200 or so individual different antibodies that have been cloned from those individuals. The antibody cloning process itself involves taking individual B cells, sequencing the antibody genes from those individual B cells after they have been through this process of cell division and mutation, sequencing those antibodies, and those sequences are represented in these slices here. Now, the black parts of these slices represent antibodies that are found multiple times within that individual. So this COV-107, this black slice represents a number of antibodies that have been found multiple times. The white part of the slice represents antibodies that were found once. Now, the interesting and important part is these lines that connect the slices. These represent antibodies that were found in multiple different people. Essentially, the same antibody sequence is found in people who have had infection and individuals who have been vaccinated. And every line here represents a common or shared antibody from convalescent individuals or vaccine recipients. So what this diagram tells us in total is that there are significant differences between individuals in terms of the types of antibodies they make, but there are also quite a lot of similarities, indeed to the extent that there are some nearly identical antibodies that are found in many, many different people. That's really quite important for understanding the environment in which the virus is replicating and learning to adapt to the human immune system. It also tells us that what we get when we vaccinate people, the type of immunity we get is really rather similar in qualitative terms to the immunity that one gets from natural infection. And that's quite important to know as well. So part of our job over the last year has been to sift through these hundreds, even thousands of antibody sequences to identify the ones that are most potent. And those are the antibodies that Rick just told you about that are now in clinical trial. Those antibodies that at really very low concentration, nanograms per mil, can neutralize infection. So as we add more antibody, we get less infection. And really, tiny amounts of these antibodies, nanograms of these antibodies are sufficient to stop infection. And that's very important from the point of view of therapy. Now, from the very earliest times that we were identifying and developing these antibodies as potential therapeutics, we began to think immediately about the issue of antibody resistance, and really posed this question: how likely is it that antibody resistance will emerge in the context of SARS-CoV-2? Now, understanding that the antibodies that we are cloning from human individuals to use as therapeutics, the antibodies that are generated in response to infection, and the antibodies that are generated in response to vaccination are all subsets of each other. There's overlapping types of antibodies. It's, I'm sure you can imagine, quite important to understand if the virus becomes resistant to certain types of antibodies, how will that affect antibody therapy, immunity to reinfection, and the efficacy of vaccines? And so, as I say, we've been thinking about this for some months now. So in thinking about how likely it is that antibody resistance would emerge in SARS-CoV-2, we do, I guess, a couple of thought experiments. So let's imagine a transmission chain where SARS-CoV-2 is being transmitted from one individual to another, going through what we call population bottlenecks when a virus is transmitted from one person to another. Usually involves a very small amount of virus, sometimes as little as one virus particle. We don't actually know what that number is in the case of SARS-CoV-2, but certainly there are a smaller number of viral variants that are transmitted than are present in an individual who has a florid virus replication in them. So the probability that in this particular individual that's marked with an asterisk, that the virus might mutate, change from an antibody susceptible population to one that's antibody resistant is dependent on a number of things. First, how accurately does the virus make copies of itself? So we know that in the case of coronaviruses, they do have an error rate. They make mistakes at a number that we can define. And we know that that number is quite a bit lower than viruses, for example, like HIV or influenza or hepatitis C virus. So what we call the error rate of the replicative machinery is quite low. However, the antidote to replication fidelity in terms of exploring genetic diversity is population size. And we know that the population sizes in SARS-CoV-2 are enormous. A single swab can contain 1 billion different viral RNA molecules. And so by doing a little bit of math, we can understand that, let's say SARS-CoV-2 makes a mistake once in every 100,000, at each position in the viral genome, there's a mistake made 1 in every 100,000 times. If the population size is 1 billion, it stands to reason that just about every possible individual mutation that can occur in SARS-CoV-2 does occur, probably in a significant fraction of individuals and certainly in the global population. Okay, so in terms of thinking about whether and how the virus has the capability of acquiring mutations, its population size is so enormous that every possible mutation that can be made has been made. So there are other considerations in thinking about how likely it is that antibody resistance will emerge to SARS-CoV-2. Okay, so in order for antibody resistance to occur, it will occur. The question is, will it be selected and will it be amplified in human populations? And so in thinking about that, we began to think about things like transmission dynamics. So in the case of SARS-CoV-2, what we know is that the amount of virus in an individual, and likely with it, the probability that they are infectious to other individuals, occurs quite soon after infection. So this is a cartoon representation of what we think goes on in an individual, but it's probably in broad terms reasonably accurate. So we have a peak of virus replication within the first sort of 5 to 7 days, sort of coincident with the onset of symptoms, with it the probability of transmission, and then the amount of virus begins to decline over the ensuing days, extending into weeks. The antibodies that are developed in response to that infection typically come up at sort of a little later than the peak of viremia. So one can imagine in a transmission chain like this, the virus quite rarely experiences selective pressure by antibodies before it's transmitted to another individual. And that likely underpins the observation that we haven't seen a lot of antibody resistance in SARS-CoV-2 until quite recently, because the virus has been spreading largely in an immunologically naive population. Now, of course, these things can change. Things like once the population, a large fraction of the population has been infected, they've made an antibody response, and then those antibodies decline. We know that that occurs in SARS-CoV-2 infection, and we know that some individuals are beginning to be reinfected. So that changes this dynamic here. It can be a situation that an individual that is experiencing infection already has some neutralizing antibody, and that antibody can in principle impose a selective pressure on that virus and enable the virus to experience that selective pressure and adapt to it. Other things like monoclonal antibody therapy, obviously we and others have combined antibodies in cocktails to try and ameliorate this possibility, but applications of antibodies as individual therapies could in themselves impart selection pressure on the virus. Also, tens of thousands of individuals across the world were given convalescent plasma therapy in instances where the plasma was not well characterized. So again, there could be sub-neutralizing levels of antibodies that could impose selective pressure on the virus, but not be sufficient to prevent onward transmission of the virus. And of course, as vaccines are rolled out, if vaccines are more or less effective, that is going to affect how the virus experiences selective pressure by antibodies. And the crucial point here is that if the virus is able to encounter antibodies in an individual such as this, but still maintain sufficiently high levels that virus can then be transmitted onto other individuals, then one can, in principle, get transmission chains like this, where an antibody susceptible virus encounters an antibody, a particular antibody, and then that antibody loses its effectiveness because the selective pressure imposed on that population has favored the propagation of antibody resistant strains. So from quite early times in the pandemic, we developed methods to try and study this problem. This is obviously something that's a little difficult to study in the laboratory. People would perhaps get a little upset if we took SARS-CoV-2 and tried to generate antibody-resistant versions of it in the laboratory. So what we did was to build a tool to study antibody resistance. And we did so by using a virus we call vesicular stomatitis virus. It's a very distant relative of rabies virus. It's quite a safe virus to use in the laboratory. It's actually used as a vaccine vector in some instances. There's a version of this virus where the Ebola virus spike protein has been inserted into it as an effective vaccine. So what we did was to take VSV, to remove its own spike protein and replace it with the SARS-CoV-2 spike protein. That gives a virus particle that is VSV, this vesicular stomatitis, this rather safe virus on the inside, but it has a SARS-CoV-2 spike protein on the outside. So it has the target cell tropism of SARS-CoV-2, and most importantly, the neutralization properties of SARS-CoV-2. The same antibodies that can neutralize SARS-CoV-2 can neutralize this virus. We also gave this virus what we call a reporter gene. It's a gene from a jellyfish that makes cells glow green when they're infected, and that gives us the ability to track virus replication in cell culture. And so the experiment that we do is to take this virus, we generate large diverse populations of this virus in cell culture. The replicative machinery of this virus has a rather higher error rate than does SARS-CoV-2. So we can represent quite a lot of diversity in smaller populations of virus. So we generate diverse viral populations. We mix those virus populations with antibodies. Those can be individual monoclonal antibodies from individual B cells, or the mixture of antibodies, what we call the polyclonal plasma, that's generated in individuals where hundreds of different types of antibodies have been made. So we take those virus populations, mix them with antibodies, and then we simply put them onto another cell culture, okay? So members of this diverse population that are antibody sensitive will be blocked from infecting these cells and lost from the population. these escape variants that we call them, antibody resistant variants, will infect those cells. They will replicate, be amplified, and then after a couple of days, just takes a couple of days to do this, we can see which viruses, which variants have escaped the inhibition by these antibodies by sequencing them. We can even isolate the individual viruses and go back and understand, did they really get resistance to the antibodies that we applied in this experiment? So I'm just going to show you some actual real data. So these are little dishes full of cells. They're about 2 centimeters in diameter, and they've got cells laying on the bottom, and we've infected these cells with about 1 million variants of that recombinant virus that I just told you about. Now, if we do that without antibody, all of the cells in this culture turn bright green, and we can see them in the fluorescence microscope. This sort of green speckly pattern is infected cells. Now, if we add an antibody, then most of the cells do not get infected because this antibody is very good at neutralizing the virus that we have made. However, if we look closely, in this area demarked by this little white square, we can find little areas of cells where the virus has infected and begun to grow. And this signifies the presence of an escape mutation, a variant in the SARS-CoV-2 spike protein that has learned to resist the effects of that monoclonal antibody. And so we've done this many times now, but one of the very first mutations that we identified by this approach is this mutation E484K. So let me just show you the experimental results. So here's the virus that we begin with. It's the VSV virus that has a SARS-CoV-2 spike. It's very efficiently neutralized by these 3 antibodies that were part of the cloning effort here at Rockefeller. Tiny amounts of antibodies can totally block infection. Now, after having done the experiment that I just showed you, growing that virus in the presence of this antibody, C121, then what grew out was a virus, that little group of green cells. You sequence the virus that comes out of that, and it's this virus, E484K. Okay, this was one of the very first mutants that we found during this approach. And when we started to publish this work about 6 months ago, I think it's fair to say that the field underestimated the importance that variation in the spike protein would eventually, the importance that it would take on. And this is a quote from one of our colleagues in the field when we began to talk about antibody resistance. I'm not worried about this at all. The dogma in the field was that antibody resistance in coronaviruses was not something that really should be particularly worried about. Now, here we are 6 months later, and it's not that infrequent that I take phone calls from journalists, and all they want to talk about is E484K. And of course, now we're quite concerned, I think, about the emergence of the variants that Rick told you about. And of particular interest is changes like E484K are appearing in multiple places in the world, particularly in the Brazil and South Africa variants that you've heard about. The UK variant originally did not have E484K, but just in the last week or so, it's been reported that there are now derivatives of that original B117 UK strain that in fact do have the E484K. And it's very likely in our view that these are mutations that are arising in response to antibody selective pressure. We're sort of emboldened in that view by a number of experiments that we have done recently. So again, using this experimental scheme where we mix the virus with a number of different monoclonal antibodies. We've now done this with a rather large number of antibodies. This table shows some of the results that we have gotten. And so what we're doing in this experiment is to do exactly this experiment, mix the virus with individual monoclonal antibodies. Those monoclonal antibodies are listed down the left side here. These are all cloned from people who have received vaccine, but as I've shown you, they're basically the same types of antibodies that exist in people who have been infected. And what this table shows are the percentages of viral sequences with the indicated mutations, these mutations across the top, basically after 24 hours of replication in the presence of each of these individual antibodies. And if we just go through the variants that come out of an experiment where we allow antibodies to select the viral variants that can resist those antibodies, what do we find? Well, we find mutations at position K417. That's exactly the same place that we find mutations in the South African and Brazilian strain. N439K, that is a variant that's actually achieved quite significant numbers of cases in Scotland initially and now in parts of Western Europe. N440, those mutations are achieving quite high prevalence in India. L452R, that is the so-called California variant. If you pay attention to the news in Southern California, you'll find that people are very worried about this particular variant in California. F456 and N460 haven't yet hit the news, but I confidently predict at some point in the not too distant future they will. E484 we've already heard about. There are a number of antibodies that seem to select for variants at this position. Another two mutations not yet in the news. N501 again is present both in the UK strain, the South Africa strain, and the Brazilian strain. And so the impression we're beginning to get is that these antibody selection experiments that we do in the laboratory are, in a sense, predicting the types of variants that are now rising to prominence in nature. This is basically the same result. It's really a confirmatory type of experiment. In this case, we actually make viral variants with particular mutations and then ask how much antibody do you need to neutralize those viruses? And if the starting virus, what we call the wild type original strain, you need rather small numbers of small amounts of virus. This is the number of nanograms you need to neutralize that virus. But if those viruses are engineered to have these resistant mutations, what's shown in red here, here, and here are the variants where you need much larger, and sometimes you can't neutralize the virus with whose spike protein has those variants. And again, K417, E484, and N501 are prominent in these types of experiments. But there are other variants that are present in nature that do also appear to confer resistance to individual antibodies that have been cloned from human individuals. Now, with Pamela Bjorkman's group, we've been trying to understand this in structural biological terms, and this is the work of Christopher Barnes and Pamela. And so what you can see here, this sort of gray cloud shape, is the receptor binding domain, the RBD portion of the spike. Down here is the spike protein, as sort of a lower magnification version. And here's the RBD. And these squiggly lines represent different antibodies that can interact with the RBD. And you'll see that each of these mutations that we think are quite important, they form part of the binding site on the RBD that's recognized by these antibodies. And so mutations at these positions really make it more difficult for those individual antibodies to recognize the RBD. Now, this all sounds quite worrying, that the virus is making mutations and it's causing really quite high levels of resistance to these individual antibodies. But there is some good news, okay? And some of the things we found lead us to think that this will be a problem, but perhaps not quite as serious a problem as the impression that I might have given to you thus far in the talk might suggest. So one of the things that we've been finding is that this process of maturation, the process that the B cells go through once they have recognized the viral protein, the viral antigen, they go through a process of mutation. The antibodies get more and more mutated, and they're selected to be higher and tighter binders to the viral protein. This seems to have, at least in some cases, quite an effect on their ability to cope with resistance mutations that the virus might acquire. So in effect, in response to the, it's not just the case that the virus is evolving, the antibodies can evolve in individuals too. And so in these two examples, what we have done, so Michelle's group cloned antibodies from individuals very soon after their infection, and then some months later, after the antibodies had acquired higher levels of mutation and had learned to bind the virus tighter. And you can see that if you plot on the y-axis here antibody potency, so the lower you are on the axis, the less antibodies required to neutralize the virus. So better antibodies are plotted lower down on the y-axis. You'll see we have the wild-type virus represented here, and then a whole bunch of variants that are found in circulating populations represented on the x-axis here. What we find is that antibodies that are cloned very shortly after infection, there are quite a number of variants represented by these green dots here, where the antibody has lost its potency. Conversely, if you take antibodies later after infection, then they are much more resilient. So mutations here that cause resistance to the 1.3-month antibody, they are much less able to avoid antibodies that have been generated after a longer period of time. Very similar example here. So the antibodies themselves can get better. They can mature with time, okay? The other thing that's quite important is that while we've focused on a particular sets of mutations, it's not the case that all of the antibodies are focused on one spot on the viral spike protein. So I've talked primarily about the RBD, okay, the receptor binding domain, this blue part here that sticks up and recognizes the ACE2 viral receptor. And we found a large number of mutations in the RBD that can confer mutations to individual antibodies. But what you'll notice from these sort of colored patches that mark where antibody-resistant mutations occur is that they occur at a large number of different spots, both within the RBD itself and also in other places on the spike protein. So every position here that's marked in red marks a position that we think is important for recognition by antibodies that can neutralize the virus. So while we analyze the effects of antibody resistance one antibody at a time, it's almost never the case that one antibody at a time will be asked to neutralize the virus. When we're immunized or infected, we make dozens, perhaps hundreds of different antibodies that all of which act together basically to cover each other's back to try to neutralize the virus. So really the issue in terms of vaccines becoming less effective and antibody resistance mutations becoming important really is a numbers game. How many different mutations does the virus have that confer resistance to how many different antibodies? And so we're beginning to understand that. And this is the sort of experiment we do where we analyze the effects of groups of mutations. So the K417, E484, and N501 mutations that you've heard us talk about over and over again. What do they do when they're all together in one viral spike protein and they're asked to avoid the whole cohort of antibodies that are generated by an individual that's either had infection or been vaccinated? And that's basically the result you see here. So every point that's represented here, we've plotted what the plasma neutralization potency, the mixture of antibodies in plasma, how good they are at neutralizing the original virus, and then this constellation of mutations that we find both in the South African and the Brazilian virus. And you can see that for most individuals, there's perhaps a small or no declination in the potency of that plasma versus the wild type virus as compared to the mutant viruses. For some individuals, there's a large change. You know, this is a logarithmic scale, so we're talking about a 50-fold drop, but that's pretty rare. For most individuals, there's a rather small change. It perhaps gets more exaggerated towards the bottom end. If you have low levels of neutralizing potency, then it looks like those plasmas are less good at coping with the mutations. And so what we anticipate is that really giving people as much immunity as possible, perhaps through booster shots, perhaps through changing the vaccine to better match circulating strains, is what the outlook will be in the future. Okay, so let me wrap up with a couple of take-home messages. So the SARS-CoV-2 spike protein elicits similar potent neutralizing antibodies in convalescents and vaccine recipients. Inevitably, antibodies exert evolutionary pressure on SARS-CoV-2 and can select for mutations that can confer resistance to individual antibodies. But the effects of those individual antibody mutations are ameliorated first by antibody maturation. The antibodies get better over time. The polyclonal nature of the antibody response, the fact that many antibodies are going after the spike at once, at least in most people. And of course, there are other components of the immune system, T cells, for example, that we strongly suspect are important, but they're just more difficult to study and more difficult to demonstrate unequivocally that they are important. All right, let me wrap up by acknowledging again the fact that this is a huge team effort. All of the work in my laboratory is co-led by my partner in science and life, Theodora Hatzianu. I'd acknowledge in particular 3 postdocs, Frauke Mücksch, Fabian Schmidt, Jiska Weisblum, who have been working incredibly hard since the beginning of the pandemic to bring this work to fruition. Other members of my lab who've contributed are listed here. As I've mentioned, Michelle Nussensweig's lab have really been key in this effort in many aspects, both of the clinical work and the antibody cloning itself. Charlie Rice and Pamela Bjorkman have also been key partners in the work that I've talked about today. Okay, thank you very much for listening.
Thank you, Paul. That was really a tour de force and fascinating. I realize that the hour is late, but I do want to take some time for questions for those of you who can stick around. Paul, one of the things I'd like to ask you about is you focused on the polyclonal sera. How about the different antibody cocktails that are approved for use or are in development for treatment?
Right. So that is individual depending on which particular cocktail that's been used. So Regeneron have theirs, Lilly have theirs, AstraZeneca have theirs, and of course Rockefeller, we have our own cocktail. In general, what we're finding is that because the antibodies that have been selected for inclusion in those cocktails have been selected to target different parts of the RBD. What in general we're finding is that the new variants take out the activity of one of the 2 components of the cocktail, but that leaves the other member of the cocktail fully potent and able to do its job. This really was the reason why Regeneron, ourselves, and others use cocktails to exactly to cope with this situation. I anticipate that over time the cocktails will be updated, particularly as we get these better antibodies that have gone through the affinity maturation process and are more resilient to these mutations.
Thank you. I should mention that, and congratulations, that the Rockefeller antibody cocktail just last week was licensed to Bristol Myers Squibb for clinical development and therapeutic and preventive use, which is a terrific achievement. There are a lot of questions that converge on the issue of how long do vaccines work and how long do you retain immunity after natural infection?
Right. So that was a point I intended to, but forgot to make. So we know that in natural infection, the magnitude of immunity, the levels of antibodies that are generated in individuals is incredibly variable. There are some people that make very low levels of antibodies, some people that make really high levels of antibodies, and that correlates at least to some extent with the severity of infection. And we suspect it's a reflection of really how much virus replicated in those individuals. More virus, more antibodies, more subsequent immunity. For vaccination, there is variation, but it's over a narrower range than natural infection, because the amount of antigen, the amount of vaccine that's injected into each person is a lot more consistent. How long that's going to last? So I think in the context of natural infection, weeks to decades, right? That's, I think I can't be more precise than that. It's going to be very variable. In the context of vaccination, I think the answer is going to be a bit more consistent, but we just have to wait and see. I would anticipate, based on the levels of antibodies that are generated in response to vaccination, that, hopefully years, okay? It really depends a little bit on how what happens as the variants play out, because as I'm sure you can imagine, protection is going to depend not just on how much antibody you have, but how well matched it is to the circulating viral variants. And that a combination of effects will determine how long that protection lasts.
Given these challenges, several people have asked about the potential and prospects for a pan-coronavirus vaccine.
Interesting. So we just submitted a grant application with precisely that goal in mind. It's quite complicated and difficult. I don't think it's going to be possible to generate a vaccine that protects against all coronaviruses. I think it might be possible to generate a vaccine that protects against all the what we call sarbecoviruses. These are the bat coronaviruses, the group from which the original SARS virus and SARS-CoV-2 came from. Plus, we'll already have some immunity to that group of viruses by virtue of being vaccinated against SARS-CoV-2. I think it'll be possible to build on that to provide protection against at least that group of viruses. How broad we can get, beyond that, I don't know at this point.
Paul, a number of people have asked about whether the vaccines protect against primary infection or just the development of symptoms or developing severe disease. Can you still be infectious after being successfully vaccinated?
So that's a very interesting, important question that I'm afraid I'm not sure I can answer with current information. Now, given the sort of the rather extreme potency we've seen with the mRNA vaccines in terms of protection from disease, it seems almost inescapable that there will be at least some protection from infection, reduced viral load, reduced infectiousness, benefits not just to the vaccinee, but also to the people that they come into contact with. Really, the best vaccines don't just protect the person who's been vaccinated, but protect everybody that they come into contact with by reducing the amount of virus replication in that person to zero, okay? I don't think that's going to be true with the vaccines that have perhaps lower diminished efficacy, or as is happening in the UK when people are given 1 out of the 2 doses of the mRNA vaccines. These sort of partially protective vaccines, while they might completely prevent hospitalization and severe disease, there is likely to be some level of virus replication, perhaps confined to the upper respiratory tract. What that's going to mean in terms of terminating or continuing transmission chains, we just don't know at this point. We're going to have to let the epidemiology tell us the answer to that.
A number of questions came in about how concerned should we be about adverse events following vaccination, and how does that track with age?
Well, I have to say I'm a virologist and I'm not a clinician. I have not been involved in administering vaccines to people. There do seem to be sort of rare allergic reactions. Nobody has yet died. They have all been treatable with things like EpiPens or Benadryl. And as I understand it, they are extremely rare. Given the numbers of people that have been vaccinated, these seem like pretty safe vaccines. When I say pretty safe, I mean pretty safe as compared to other vaccines that are extremely safe. So they're I would say just as safe as any vaccines that you've been used to.
Yes, I think it's fair to say the number of severe reactions have been quite limited. Most people who have had them have had a history of anaphylaxis to other vaccines or other agents. There has been some interesting data recently on people who have been previously infected who then get vaccinated, having more potent immune reactions to the virus. But again, those have generally been easily managed. Another question that has come in is, what are your thoughts about the mRNA vaccines versus the other, adenoviral or protein-based vaccines? Any reason to suspect that there will be differences in efficacy or duration of effect?
So, not really. I think you have to take each vaccine sort of as it comes and basically follow the science. It's hard to say that a protein vaccine is better than an mRNA vaccine or vice versa, or better than an adenovirus vaccine. They all have their sort of strengths and weaknesses in terms of how easily they're delivered. But you can't really say just knowing what kind of vehicle it is, whether it's going to be a better or worse vaccine.
So if I understood your closing comments about the impact of these variant viruses, my impression is that you think that there will be more people who develop symptomatic disease than compared to people who don't have these variant viruses, but that perhaps the incidence of severe disease where people end up in an ICU and at high risk of dying will not be completely attenuated, and these will still continue to be efficacious vaccines. Is that a fair assessment of your view?
That's fair. The caveat being that I don't have a crystal ball. I think it's true to say at this point that even in situations where the vaccine has been the original strain and the infecting virus has been one of these new variants, nobody has yet died in that situation. So it's clear that there's still, in every case that has thus far been looked at, protection from the severest disease and death. Now, we obviously hope that will continue. It may very well be that the vaccines need to be updated to maintain that situation. Where the uncertainty is at this point, I think, is whether that degree of mismatch between the vaccine and the circulating virus will enable more transmission chains to continue, whether it'll enable sufficient levels of virus replication and really mild disease to be propagated through the population. That's really important for achieving things like herd immunity. Okay. Does the vaccine not just protect you, but other people? That's a situation that we really would like to get to, but you really do need a vaccine that squashes virus replication enough that you're no longer a transmitter. That's sort of a key distinction, I think.
And my last question, seeing the multiplicity of mutations that are emerging on these resistant viruses, there's been a lot of speculation and in a couple of cases documentation that they may be coming preferentially from people with compromised immune systems where you can mount an immune response but can't clear the virus, and so the virus keeps getting multiple shots on goal to evolve in a single patient. How do you view this, and do you think there's an effective response that we should be making to take special precautions with people who have compromised immune systems?
Yes. One could argue that at this point it might be closing the door after the horse has bolted, but yes, I have seen a couple of reports where there have been, I would say, impressive amounts of sequence evolution in single individuals who have had compromised immunity and have been treated with poorly characterized convalescent plasma, exactly the sort of situation that I outlined, where you're putting enough selection pressure on the virus to allow it to continue to propagate and adapt to the antibodies that are thrown at it. Exactly what we do in the laboratory as we're studying this process. Now, on the flip side, if you have a sufficiently large population of people who have immunity that's waned to a level where they can be infected but can still propagate the virus, you have effectively the same situation, right? So whether the evolution occurs in a single individual or a population of individuals who have diminished immunity is sort of moot. It's going to be, I think, difficult to trace these variants back to their source, and I think it'll be impossible. But to answer your question directly, that yes, I think individuals who have compromised immune system and persistent infection should be treated quite cautiously in terms of isolation, for example.
Great. Well, there are many gearheads who have stayed with us all the way to the end, and we could go on for a very long time, but I think it's time to draw this session to a close. I want to thank Paul for a spectacular talk, incredible work. Congratulations, and thanks for joining us today. For those of you who have been joining us for these virtual discussions, we will continue to keep you informed in the coming months. Your philanthropic support has been and will continue to be vital to our continued success to combat COVID-19. Please see the next slide for more information about how you can contribute. We invite you to continue visiting our website for more information, as well as for videos of our past sessions. We'll also be announcing future webinars on that page. Thank you once again for participating today. Have a good evening.